Okay, so let's move on to our physical foundations. As we noted, organisms must perform energy, make energy, and make energy transductions to stay alive. They don't exist in equilibrium with their surroundings. Living organisms are always in a dynamic, steady state. So even though they might seem like they're at equilibrium, this steady state is dynamic. There's always some interchange between the environment and themselves. This is what we need to do to stay alive. Energy coupling allows living organisms to transform matter into energy. So by doing these transformations, we can take things like food and then make it into energy for ourselves. We'll also talk about biological catalysts. This is a physical component of energy transduction. We use catalysts to reduce our energy requirements for reactions while offering specificity. We also have a battle with entropy, and I'll review kind of those ideas and concepts. The entropy of the universe is always increasing, or it tends towards increasing, and creating and maintaining order within an organism requires work and it requires energy. So again, this is just kind of a diagram to show you the types of energy that organisms perform to stay alive. We're going to be talking about this idea of physical transformation of energy. And we see it in, for example, the transference of sunlight to different molecules, making molecules within a plant, or chemical transformations. This is where we take things like chemicals and produce mechanical work within an organism, or we make electrical gradients. That sounds weird, but an electrical gradient is how our neurons work. We have to actually transfer electrons, right? That takes work, and we have to start with something like food and then turn it into an electrical gradient. Or what if we just need to make heat? You need to stay warm, right? Organisms often produce heat. Mammals produce heat to stay warm. Or even things like metabolism. I've been talking a lot about turning fuel into energy, but metabolism produces compounds, right? We make CO2, we make water, and also we take simple compounds and then we put them together. So we make DNA, we make proteins. All of these are energy transductions that we need to do in order to stay alive. And we talked about speeding up reactions as well. Not only the ability to accomplish reactions, but our ability to control those reactions. So different ways to control reactions is through things like higher temperatures. So the stability of macromolecules is a limiting factor here. So sometimes we can't just heat ourselves up to speed up a reaction. You can use something like higher concentration of reactants. This is indeed something that is performed within organisms, but it is costly because there you have to just make a lot of starting material to utilize this as a way to speed up a reaction. That means you need to be taking in glucose all the time so that you had a ton of glucose so that you could push it to make other things. Changing the reaction by coupling it to a fast one. Now, this is something that we as organisms utilize often. Many organisms, not just focusing on animals or humans, utilize coupling reactions. This is again by taking a reaction that is favorable, coupling it to a reaction that's not favorable, and ultimately getting a reaction to occur faster. And another way to speed up reactions, as I noted, is through catalysis. That's by lowering the activation energy. We'll go through this a little bit more in a second. But again, this is universally used by living organisms. Again, across the board, catalyzation, enzymatic catalyzation, in fact, is used by organisms. So to remind you about bioenergetics, how we're going to talk about energy transformation in organisms is still by using these very basic physical equations. When a chemical reaction occurs at a constant temperature, the free energy change, that's the delta G, is determined both by its enthalpy, randomness, right, and its entropy, entropy is randomness, delta S, and its enthalpy, delta H. So delta H, again, reflecting kinds and numbers of chemical bonds and interactions broken and formed. And then the entropy, delta S, is the system's randomness. So these two components, bonds broken and formed, and the randomness of the system, will ultimately decide the overall energy change of a reaction. So it will decide if it's going to be a favorable reaction or a non-favorable reaction. Remember that things like synthesis of complex molecules and other reactions that we'll be seeing, they require energy. That means that they are endergonic. This is a non-favorable reaction, does not occur naturally on its own, and it takes energy to make it occur. A reaction could be thermodynamically unfavorable. That's where this delta G is greater than 0. So this is something like creating order, right? If you're going against entropy, this is usually something that requires work and energy. Or some metabolic reactions might be too high of an energy barrier. Again, this is a delta G greater than 0. The breakdown of some metabolites releases a significant amount of energy. So some of these reactions are exergonic. They actually go, they happen without an input of work, and they in fact release energy instead of taking energy. Things like ATP, NADH, and NADPH, these are molecules we'll be seeing and talking about this semester, can be synthesized using energy from sunlight and from fuel like sugar, and their concentration is higher than their equilibrium concentration. So these are readily available, they can be synthesized, and when they are broken down, they release energy. So what do? We couple these together with those reactions that I brought up, things like building complex molecules or some other metabolic reactions that are unfavorable. We couple them with reactions that are favorable. So this is like taking a high energy molecule, that's what we call it, ATP. The reason we call it high energy is because it releases energy. Chemically, it is exergonic. And we react it directly with the metabolite that needs activation. These are words we're going to be using, the reason they're in quotations here, so you get familiar with these words and what we actually are meaning chemically. We're taking a molecule that releases energy and we're directly reacting it with another molecule that needs energy to proceed to its final product. This coupling we usually call activation. Another concept, physical concept that is talked about a lot during biochemistry is equilibrium. So we've talked about things being energetically favorable, endergonic or exergonic, and how we couple reactions. We also talk about the tendency of reaction to go to completion based on the equilibrium. Equilibrium is still tied to something being exergonic or endergonic. I'll show you that in a second, but remember how we set up an equilibrium equation, how we talk about equilibrium. So for example, if you have some reaction, any molecules, now we can be talking about complex large molecules that are reacting. It's still a chemical reaction. Let's say we have some reaction of A that reacts with B to make C and D. To write an equilibrium constant, that's called Keq, you would set up your equation as the concentration of the products, and notice they're raised to whatever the balanced number is in front, over the concentration of the reactants, again, raised to the mole number that is in front of that species. This gives you the equilibrium constant. The reason we care about equilibrium constants is because they are easy to measure. Measuring if something is endergonic or exergonic, actually measuring it is very complicated. Instead, we can experimentally measure the equilibrium because it's just based on concentrations. So if we can measure equilibrium, we can also determine the delta G, how favorable a reaction is, if it's endergonic or exergonic. And the way to do that is by using this equation shown here. Delta G is equal to negative R. R is a constant. T, that's temperature, times the ln. This is the natural log. So remember how to do logs and natural logs. The natural log of whatever your equilibrium constant calculated was. So Keq, this is based on concentrations that you have. So if you know the concentrations of a reaction, you can calculate your Keq, and you can also calculate if it is a energetically favorable or non-favorable reaction. Generally, when Keq is greater than 1, it's bigger than 1, the delta G is large and it is negative. Remember, we have this negative symbol here. So if we have a big Keq, that means delta G is large, it's negative, and remember that means that this is a favorable reaction. This is going to make energy. When Keq is small, delta G is large and positive. And so in this case, this is going to be an endergonic reaction. It's going to require energy to proceed. And if you go back and think about equilibrium, this kind of makes sense. If we have a reaction here, and we make a lot of product, we made a lot of C and D, we don't have a lot of A and B left. So if we measure the concentrations, we had a reaction, we put A and B together, and in the end, we measured our concentration, and there's a ton of C and a ton of D as far as concentration. and there's not a lot of A and B. If we put that into our equation, let's just say this is 2 and this is 1. 2 to 1 between the two of them. I'm really simplifying things down here, but just as an example, you get the idea that when we put our products over our reactants, you get a bigger number the more products you have, right? Products is on top, bigger that number, the bigger the KEQ. What did I just say? When KEQ is big, delta G is negative. So big KEQ, lots of products, that means it's exergonic. Again, that makes sense. You made a lot of products. That means your reaction is driving, it's going forward, it's making energy, it's making products. If you have the opposite, if you do a reaction and it just kind of sits there, it doesn't really make any products. You have way more reactants than you have products when you measure them. Plug that into your KEQ. This is a small number now, right? It's a fraction, it's only 0.5 versus 2. When KEQ is small, less than 1, delta G is positive. That means it's exergonic. Again, makes sense. You're not making a lot of products. Your reaction isn't driving forward. That usually means that your reaction needs energy to be driven. So this is how we kind of combine those ideas of equilibrium. We can measure equilibrium, and we can use it to understand if something is going to be endergonic or exergonic. You can also do the opposite. If you know something is exergonic, you can probably predict that its KEQ is going to be larger than 1. It's going to be driven towards the products. So here's an example of looking at coupling those reactions, what it actually looks like on an energy diagram. You'll be seeing these energy diagrams. So this is the completion of the reaction going from starting to finish. And we're measuring the delta G, that change in energy as a reaction goes. So if you have a change in energy that is high like this, a positive delta G, it's like going up a hill. It's hard to get there. It's hard to go from low energy to high energy. And so this is non-spontaneous. It's a endergonic reaction. So something like taking glucose and adding a phosphate to make glucose 6-phosphate takes work. But we also know that this reaction up here, taking ATP and breaking it into ADP and phosphate, this is one of those coupled reactions, clue here. And you'll be seeing ATP a lot. Look at its energy diagram. It goes downhill, right? It goes from high energy to low energy, and it is a big drop. Just like riding your bike. This is easy to do, right? Going down the hill's the fun part. It's easy, you just sit there. This is an exergonic reaction. So if we combine these two, we combine going down a big hill and then having to come up a little hill, our overall energy diagram would look something like this, right? So it's still going to be easy to do. It's not as big as a drop of the reaction of ATP versus ATP, but this reaction is still going to be going from high to low energy. Now we're seeing a visualization of those energy diagrams, but you can also calculate it using numbers. The final energy is just going to be adding the delta G of your first reaction with the delta G of your second reaction. So you'll have a positive delta G and a negative delta G that you add together in this case. And overall, you still will have a negative delta G at the end, something that is exergonic. So remember, we'll be practicing calculating our delta Gs here. Now, another thing we talked about in bioenergetics, how we make a reaction go faster. We just talked about coupling, what that looks like, the equations we use when talking about coupling reactions. But we also talk about catalysis. So remember, a catalyst is a compound that increases the rate of a reaction, but it does not actually change the reaction, right? The catalyst is not involved in the reactant or the product. It's not one of the reactants or the products. It just increases the rate of the chemical reaction. And it does this by lowering our activation energy. They don't alter the delta G. So in an example like this, our activation energy is this little hump you have to get over. Notice all of these have a hump. This is our activation energy. So by lowering that piece, it makes the reaction a little bit easier to do. Now, again, it doesn't alter the delta G. That means the starting and the final place you have to get to, starting place, final place, those are the same. But that hump, the little activation energy, is lowered. So for example, if this was what you had to do to go from reactant to product, a catalyst would do this. Gets rid of that activation energy, but still the change from reactant to product is the same. The way that we do catalysis in biology is through things called enzymes. These enzymatic catalysts offer, again, acceleration of a reaction in mild conditions. They're highly specific towards certain components of a reaction, and they allow for regulation of a reaction as well. So sometimes if you don't want a reaction to proceed fast, you can limit the catalyst. So again, here is an example of what that enzyme lowering of activation looks like in order to increase a reaction rate. Here, in this case, we have a pattern that's going from high energy of reactants to low energy of products, but the idea is still the same. We're lowering just that activation energy barrier that you have to go through to then get to your final products. We can also serially do these reactions enzymatically. So when we have a series of related enzymatically catalyzed, so reactions that are catalyzed using an enzyme, and they're in a pathway, we just call this a metabolic pathway. And sometimes it's called a signal transduction pathway. Generally, we're calling it a pathway. That's some series of reactions where we go from A all the way to F, but in between, right, we have each of these steps, A to B, B to C, C to D, etc. And each of these steps uses an enzyme. They use a catalyst in each step. This is called a pathway, and we'll be seeing a lot of pathways in biochemistry. Your body utilizes pathways all the time. When they're a pathway that has small molecules like glucose to fructose to galactose, those are all small sugars, and those are going to be what we call metabolic pathways, small molecule pathways. This is just learning vocabulary here, and I'll be saying this a lot throughout the semester. When you have something that utilizes proteins, bigger molecules, not small molecules, same concept, A to B to B to C to C to D. This is what we call a signal transduction pathway. So this is just protein. The only difference is that we're talking about proteins interacting with proteins instead of small molecule with another small molecule. And as noted, pathways can be controlled in order to regulate things that are happening in the body. So one way to regulate these pathways is by regulating the amount of enzyme. I said that before, right? If you control how much enzyme is around, then you're controlling if that reaction can be catalyzed or not. If it can't be catalyzed as much, it's not going to go as fast. So by limiting enzymes, we ultimately control a pathway. And this is something that you'll be seeing. Mostly this is in Biochem 2. We'll be talking about metabolic pathways and feedback loops and how they control each other. But this is just a nice example of that idea of what a catalyst is, how it works, and how our body can use it to control and regulate steps